Thank you very much. So I acknowledge that we meet today on the lands of the Turrbal, Jagera and Yagara peoples, and that I conduct my work on the lands of the people of the Kulin Nations, and I pay my respects to their elders, past, present and emerging. And so I thought that I'd be fainting with terror by the time I got up here today, but I'm absolutely thrilled to be up here this morning. I've been coming to HBM for almost 20 years, and my first HBM conference was actually the 2008 one held in Melbourne. And I'm really excited to tell you about our lab's work in understanding dynamic glucose metabolism and the metabolic networks of the brain. So I'd like to start with one of my favourite quotes to set the scene. And this quote is from Lachlan and Sejnowski, where they state that the brain is a biological network that's organised to maximise the information encoded and minimise the energy expended. And so I think that we as a community do quite a lot of excellent work to understand the structural and functional architecture of the biological network that is our brain. And I also think that we do a lot of really beautiful work to understand how information is encoded and transferred across this brain network. I think that we do a little bit less in understanding how energy shapes the functional architecture of our brain network, and this is what I'll focus on today. So we've known for about 130 years that the brain is a big network, and so we've known this since the establishment of the neuron doctrine in the late 1800s. Quite early on, it became clear that a cross-correlated or coherent neuronal activity might form the basis for information transfer in the brain, and this concept of functional connectivity emerged. So it's quite an old concept now. The importance of glucose in shaping the functional architecture of the brain became quite clear as well in the 1940s, and particularly in the 1960s with the development of the deoxyglucose PET method by people like Ketty and Schmidt and Sokoloff and people like that. And from this work, we know that the supply of energy, the energetic costs of maintaining the brain network and transfer information is a central factor in shaping the network of the brain. A reliable and scalable supply of energy is critical for the maintenance of brain health. And it's this scalability that I'll be talking a fair bit about today. And by scalable, I mean that we're able to up and down regulate our energy metabolism on demand. We know that if you have a disruption in your ability to receive energy to your brain or to use it dynamically, sets off what's been called as a brain energy crisis. And so if you have an acute and abrupt disruption to the metabolic supply of your brain, you will lose consciousness and you could die. But if you have more subtle and chronic disruption to the metabolic supply to the brain, you'll start to encounter a number of neurological conditions. And so we see this in conditions that we consider as classically metabolic, like diabetes and polycystic ovary syndrome. But you also see disruption in the energy supply and use in conditions like Alzheimer's disease and Parkinson's disease, but also in things like schizophrenia and mood disorder. And disruption of the metabolic system of the brain is actually very ubiquitous across psychiatric and neurodegenerative illnesses. We see signatures of the importance of energy metabolism in the functional architecture of our brain. And so here we can see kind of this classical functional network where we can see characteristics that we're all used to, so things like modularity and the existence of hub regions that have higher levels of interconnectivity than non-hub regions. Whether or not a brain region is a hub or not has implications for how it uses its energy budget, with hub regions using more of its energy budget in synaptic transmission than non-hub regions. And we can see this when we start using macroscale neuroimaging, with the energy metabolism of a brain region increasing linearly with its region degree, and the energy metabolism of those connector hubs being higher than provincial hubs and non-hub regions. And so you might be wondering, well, what is the form in which we receive energy in our brain? Under normal circumstances, glucose is the primary source of energy in our brain, and neurons are heavily reliant upon glucose for generating postsynaptic potentials. At least on a conceptual level, brain glucose uptake takes place on a fast timescale, and so in around the timescale that it takes for a single synaptic transmission event to occur, it's likely that you're metabolizing 1 molecule of glucose per synapse per millisecond. So that's conceptual. Whether or not we can actually measure it that quickly is another question. So glucose metabolism is therefore essential for information transfer in the brain. It plays a central role in shaping the anatomy and functional topology of the brain, and it's also critical for the maintenance of brain health and function. So today I want to tell you a story about recent developments in measuring and understanding the dynamic use of glucose in the brain, so what we in our lab call glucodynamics, and how we've been using these advances to understand the metabolic network of the brain. Finally, I'll discuss what happens to our dynamic use of glucose and the metabolic network of the brain as we age. So we'll start off with glucodynamics. So if you want to measure glucose uptake and metabolism in the brain, your method of choice is FDG PET. And FDG is a radioactively labelled glucose analogue that accumulates in metabolically active structures. And so in the brain, it's usually accumulating in the postsynaptic terminal. Now, a component of the bold fMRI response that we all know and love does include glucose metabolism. But again, as most of us know, the bold response also includes vascular and oxygenation components, and they're all kind of interacting in a way in which we don't fully understand. And so that means that when we're using bold fMRI, we have a semi-quantitative index of neuronal function. Now, thanks to many decades of really beautiful physiological and pharmacokinetic work, we have a pretty good understanding of how FDG PET is taken up to the brain and is metabolized, and how this relates to the underlying glucose metabolism. And so this means that FDG PET is a more direct and quantitative index of neuronal activity than bold fMRI. Next slide's not coming up. Thank you. So while FDG PET is a really powerful and quantitative neuroimaging tool, the way in which we traditionally use FDG PET actually does away with some of the temporal information that we're interested in when we're interested in understanding brain function. So most traditional PET approaches will acquire a static image in the sense that you get a single image across the scan duration. And so a scan duration might be 10 to 30 minutes under typical circumstances. And so that means that our effective sampling rate, or what we would call TR in MRI, is the same as the scan duration. And so as a traditional PET acquisition can be truly static, where no temporal information is acquired at all, or it could be a dynamic acquisition, but what ends up happening during post-processing is that you use that dynamic information to summarize the rate of glucose metabolism across the entire scan. And so that means that regardless of whether or not you're taking a truly static or dynamic acquisition, you're still ending up with that single quantitative image at the end. And so we were interested in whether or not we can use the temporal information that is clearly evident in the FDG PET signal. So this image off to the side here is actually me being injected with FDG. And as you can see, there's a lot of temporal information there that we can use to understand the brain. So can we extract that and analyze it to understand glucodynamics? And so the short answer is yes we can. About a decade ago, this approach called the functional PET or FPET approach was developed by the group at MGH. And the idea here is that if you maintain a constant plasma supply of the radio tracer, you can start to index subtle and dynamic changes in glucose uptake, either in response to a task or during rest. And so in the decade since that time, both my group and other groups in the world have been continuing to develop this functional PET approach. And we're now at the point where we have a temporal resolution in the order of 16 to 30 seconds. And so as you can see here, the functional PET approach has resulted in a step change in the temporal resolution that we can obtain with this data, reducing it from 10 to 30 minutes down to the second level. Just for reference, all the data that I will show today has been acquired on a simultaneous PET MR scanner. However, it can be acquired on a traditional PET CT scanner. You don't need the MR. And all the data I'll show you will also be reconstructed with a 16-second frame duration. So we've seen in the literature that, that frame durations are reducing to the sub 10-second level. However, when you start to get to that level, the SNR and the computational demands are really, really huge. So for reference, it took us 8 months to reconstruct 90 subjects with an effective TR of 4 seconds. And so the data is huge. So 16 seconds for us is a good compromise. Functional PET does seem to be quite reliable with test-retest reliability in the order of that we obtain with ASL and outperforming fMRI. And so it's quite a powerful approach. So the first question is, well, what are the characteristics of this glucodynamic signal, and is it a useful measure? And so you might just be interested, well, what does the time series look like? And so this is an example time series here with a frame duration of 16 seconds and with the simultaneously acquired fMRI signal with a TR of 1 second. And so here you can see, yes, there is temporal information here in the time series. So how can we characterize this? Well, one way to be able to characterise this signal is by thinking about the variability and the complexity of the glucodynamic signal. And so we do this quite a bit with the hemodynamic signal in fMRI. So we measure things like the dynamic range or the standard deviation of the hemodynamic signal. And when we do that, we find that people who have a more variable hemodynamic signal tend to do a bit better cognitively. If you want to understand the complexity of the glucodynamic signal, the way that you do so is by looking at measures like signal entropy. And here the rule of thumb is that a higher entropy indicates a more complex and irregular signal, which means that the brain can support a larger repertoire of potential states. And so with hemodynamic entropy, while the rule of thumb is that more is better, it does turn out that it is a little bit more subtle than that, with variations across the cortex in the relationship between higher entropy and cognition. However, it is a useful rule of thumb, and it's a rule of thumb that we'll use here. What does the glucodynamic variability and complexity look like? So these results are really hot off the press and are not yet published, and we're still exploring this data. But you can see that the functional PET and the functional MRI signal varies quite a bit over the cortex, with glucodynamic entropy being highest frontotemporally and variability being the highest frontoparietally. With regards to the fMRI, we can see that the hemodynamic variability and entropy is highest in parieto-occipital regions. And so what we were interested in was, well, are these parameters useful in understanding the cognitive function of the individual? And you can see here that the relationship between glucodynamic entropy and variability is significant and does outperform that of hemodynamic variability and entropy. And actually, when we look at these in a larger regression model, the strongest prediction of cognition was the glucodynamic variability and complexity. And so, as I said, we're still trying to understand these results a little bit more, but we think that this is indicating to us that the characteristics of this dynamic glucose uptake signal is useful for understanding the brain and cognition. And so to summarise, if you want to measure glucose metabolism in the brain, you use FDG PET, and there have been a lot of developments over the last decade in how we acquire and use FDG PET imaging, including this functional PET approach. So our group have used this functional PET approach to understand time-varying glucose uptake in the brain. And while I haven't covered them, there are a lot of technical issues that are still being addressed with this FPET approach. However, with the emergence of next generation PET scanners, like these highly sensitive large field of view scanners, will be helping with some of these issues as we move forward into the next 5 years. At the moment, we think that glucodynamic signals share similar variability and complexity properties as hemodynamic signals, and they might outperform fMRI for the prediction of cognition. And so I'm just going to have a quick drink. Okay. So, what about the metabolic network of the brain? And do these glucodynamic signals form some sort of coherent metabolic network? So, the first thing that I want you to know is that metabolic connectivity is not new. Network analysis of FDG PET data actually predates that of fMRI by at least 10 years. The field of PET connectivity in the 40 years since this first paper has been a small but active field of research, both here but also in the brain PET community as well. And so there's been interest in understanding whether or not metabolic connectivity might be a biomarker for disease. And this is quite interesting to people because FDG PET is actually one of the only macroscale neuroimaging tools that we use routinely in the clinic to diagnose people. And so people have been interested in understanding, okay, can we use metabolic connectivity as a biomarker for something like Alzheimer's disease? But a little bit of the elephant in the room here is actually these connectivity matrices are not estimable at the individual level. You can only estimate them across subjects because you do just have that single image per subject. And so we've argued that if you can't estimate a connectivity matrix within a brain, then we should be thinking of this a bit more like a cross-subject covariance measure and should be trying to be able to measure connectivity within a brain so that then you can use it as a biomarker. And so that was what we did in our first work in this area, was effectively say, well, can we use the functional PET approach to estimate a metabolic network within a brain? And so with fMRI, I think you're all familiar. The way that we would estimate a network is that we would extract a hemodynamic time series across all the regions of the brain, form some kind of mass cross-correlation within a subject. Every subject has their own little connectivity matrix that we then enter into a grand average. And so we effectively just copied this approach with functional PET data. So here on the left, you can see that the approach that's taken with a standard PET approach is quite different because you don't have that time series to be able to capitalise upon. And so in this early work, we hypothesised that we would find a modest similarity between the functional PET and the fMRI network. And we thought that primarily because a component of the BOLD response does include glucose metabolism. We also expected to see moderate similarity between the functional PET and the standard PET covariance matrix. And the reason why we expected this is because it's the exact same data. It's just that we've chopped it up and analysed it in a different way. And so it's the same data, so we thought that it was reasonable to expect that there'd be some level of correlation there. So what did we find? So you can see first off that the matrices that we obtain are very different between the three approaches. And just focusing on that FPET connectivity matrix in more detail, one of the first things that you'll see is the signal-to-noise ratio is really quite low here. So this was our first attempt at doing this, and in the time since we collected this data, we have improved our acquisition and processing a bit more, and probably more than double the effect sizes that we get. However, it's important to note that it's still a low SNR approach. The other thing that you'll notice is that the FPET matrix seems to be predominated by frontoparietal connections. And when you look at the network analyses that you can see in the green plots underneath there, you can see that the FPET connectivity matrix seems to be predominated by high connectivity in higher order functional networks. And at least qualitatively, if you compare that to what we're getting with the fMRI, that's quite similar, right? Like there's high interconnectivity in higher order functional networks and lower in sensory motor networks. Shifting our attention to the standard PET approach, you can see that there's a higher level of interconnectivity in that matrix, particularly with the sensory motor networks as well. And so at least on a qualitative level, we can see some similarities, but we can also see some differences here. Now, at the time when we derived these results, I did think it was interesting that we were seeing primarily frontoparietal activity. However, I tend to be biased because I like executive functions that are frontoparietally driven, and so I'm just always kind of happy to see those regions come out. However, in recent years, the significance of this has become a little bit clearer. There's a beautiful work by Valentin Riedel in Munich, where they showed that these higher order functional networks have a higher proportion of highly energetic brain regions associated with them. And so this is starting to make sense that we're seeing more connectivity in regions that are more metabolically demanding. So when we compared the three matrices, what did we find? We used a number of different approaches for comparing these matrices, but I always go back to the very first one that we ever ran just because a simple correlation is always really easy to understand. And so if we look at the FPET and fMRI correlation on the right there, you can see that we saw a correlation between the networks across the brain in frontoparietal and temporal regions. And the effect sizes were moderate. And so this is consistent with our hypothesis that we would see some moderate similarity between these two matrices. And the data that we presented in the paper is consistent with this as well. Interestingly, when we compared the functional PET and the standard PET networks, it was really surprising to us to see that that there was virtually no relationship between the two networks. So this was surprising to us because it is the same data. We've just estimated the connectivity profile in a different way. And so these results have been replicated using very different approaches by independent groups in the time since then. And so on the basis of these results, we set off a little bit of a firecracker and said that if you're using a cross-subject approach, that we should be considering those results as metabolic covariance and not necessarily connectivity. And so for us, the standard PET covariance approach is not a valid index of what's happening in the metabolic network within an individual brain. So we now have a way to map the metabolic network of the brain within a subject for the first time. Does this tell us anything about the brain that we didn't already know and couldn't already obtain with our usual tools like fMRI? So in our very first exploration of this question, we simply just looked at how metabolic connectivity and the simultaneously acquired functional connectivity predicts cognition. And we did find that both of the matrices did predict unique variants in cognition, with the metabolic connectivity network predicting primarily executive function, so things like proactive and reactive control, whereas the functional connectivity matrix was predicting a broader range of cognitive functions. So things like proactive and reactive control, but also memory and depression as well. And so this was our first little bit of insight that perhaps the metabolic connectivity is providing unique but complementary information about the brain. So stepping back a bit again, you might be thinking to yourself, okay, but of what use is glucodynamics and metabolic connectivity? You know, we've had this really powerful way to understand the metabolism of the brain using classic metabolic rates of glucose developed by Sokolov et al. In the 60s and 70s. Are we actually getting anything more useful about the brain than what we could obtain with classic measures? And so in this set of results that I'm going to discuss now, these are the questions that we were exploring. So we explored this by looking at the relationship between metabolism and cognition. And the questions that we're asking was, well, what's the important predictor of cognition? Is it the size of your energy budget? Is it the way in which you use energy, or is it the metabolic network parameters? And so again, this is not a new question. People have been asking these questions for decades as well. And we were thinking, well, if we think about resting metabolic rate of glucose as somewhat analogous to how we think about resting state functional connectivity, so as a way of forming the fundamental framework of the brain upon which exogenous activity then builds, then we'd expect that the resting metabolic rates of glucose should be related to cognition. And people have been asking this question for decades. And at least by the mid-1990s, evidence in favour of this hypothesis was equivocal. So we'd have some beautiful results by people like Cheryl Grady, who would show no relationship between resting metabolic rates of glucose and cognition. But then you'd see some other similarly beautiful research that was showing a relationship. And so by the mid-1990s, people were saying, well, look, the result doesn't seem to be clear, but we think that probably resting metabolism does have an impact on cognition, and maybe a more intelligent brain is just using its energy budget more efficiently. Now, in the decades since then, we've had new technologies like PET-MR come out that have a better spatial resolution. We know more about the functional networks that are present in the brain that we didn't know in the 80s and 90s. And so by using these new technologies and these new knowledge, we had another look again at whether or not resting metabolic rates of glucose are related to cognition. And we saw that yes, in these functional networks, we do see a modest relationship with cognition. And so returning to the original question then, well, is it the size of the energy budget? Is it the resting metabolic rate of glucose that's important for cognition? Is it the way in which it's used? Is it the variability in the glucodynamic signal? Or is it the efficiency of information transfer across the metabolic network that's important for cognition? And so to do this, my PhD student Hamish used path analysis, and he did this at both the whole brain and the network level. And what's interesting is that if he included a direct path from resting metabolic rate of glucose through to cognition, every model that he tested had a poor model fit and low and small effect sizes. And so strikingly, there seems to be no direct influence on resting metabolic rate of glucose on cognition. However, resting metabolic rate of glucose does influence cognition indirectly through both glucodynamic variability, but also its coherence into networks, such that a larger resting glucose metabolic rate is associated with more variable glucodynamic signal, higher metabolic network local efficiency, and lower metabolic network global efficiency. And all this goes through to cognition to be related to improved cognition. And so this is indicating to us that it's the glucodynamic variability and the integration and segregation of the metabolic network that's important for driving cognition. And when we looked at the network level, what was quite interesting was that for almost for all networks that we tested, we did still see that indirect pathway from metabolic rates of glucose through the variability of the glucodynamic signal and through to improved cognition. And the efficiency metrics of the metabolic network were important for the control and the ventral attention networks only. So the local efficiency of the control network, improved local efficiency in that network was associated with improved cognition, and lower global efficiency of the ventral attention network was associated with better cognition. And so taken together, it seems that it's not the size of the energy budget per se that's important for cognition, but it's the way in which it is used. And so this is intriguingly similar to the speculation that we were seeing in the mid-1990s, that the more intelligent brain works efficiently rather than hard. And so taken together, we think that metabolic connectivity does yield complementary but unique information about the brain, and it seems to be really predominated by connectivity in those higher order functional networks. And while the majority of the energy budget of the brain is spent in maintaining the resting state, it's not the size of this energy budget per se that influences cognition, but the dynamic range of the glucodynamics and its coherence into networks that's important. So, in the last 8 minutes that I have, I'm going to talk about how this changes across the lifespan. So from a maturation perspective, cerebral glucose metabolism is really interesting. By the time you're hitting your 80th birthday, your brain is using about 3/4 of the amount of glucose that it was using when it was 18. And if you're on a healthy aging trajectory, your glucose hypermetabolism will be primarily frontal temporal. But if you're unlucky enough to be on a trajectory towards Alzheimer's disease, then your hypermetabolism will be more posterior. Younger brains have got a high glucodynamic range, and its glucodynamic signal is unpredictable and complex. And so this supports a highly adaptive metabolic framework that can adjust to shifting tasks and demands. By comparison, older adults have got a lower glucodynamic range and a less complex glucodynamic signal. And so this results in a less adaptive and less flexible metabolic network. What about metabolic connectivity? Well, we know quite a bit about functional connectivity. We know that as we age, the brain becomes less modular, it becomes more integrated, less segregated, and less efficient. So do we see the same thing with the metabolic network? So here are our metabolic networks for our younger and older adults. The first thing you'll notice is that these matrices look quite a bit different to the previous ones I showed you, because here we're using the Schaefer Atlas. But if you take a look here, you can visually see that it seems like older adults have got lower metabolic network connectivity than younger adults. And when we have a look at this at a network level, this is exactly what we see. So older adults are showing lower within and between network metabolic connectivity relative to younger adults. And I was just interested in which parts of the brain this is really happening, because it can be hard to see this in the Schaefer parcellation. And so we just had a quick look at the anatomical parcellation, and these do seem to be primarily frontotemporal, which is consistent with patterns of glucose hypermetabolism that we see in meta-analyses. As I said, we always have functional connectivity results because we use PET-MR. And so we had a look at what was happening with the functional connectivity. And at least here, the first thing you can see visually is that the functional connectivity matrices look like they're more highly connected in older people than younger people. And this is exactly what we see when we kind of like start looking at networks. The older adults have got higher functional connectivity but lower metabolic connectivity relative to younger adults. And so this is the first set of results that I've shown you today where the take-home message isn't complementary and unique. So there's an exact opposite effect happening here. And so why is this? And so at the moment, it is speculative, but what we think is happening here is that we do see in ageing a relative level of neurovascular uncoupling in older age. And so this is not a catastrophic neurovascular uncoupling, it's very subtle. We see subtle changes in the relationship between cerebral blood flow and glucose metabolism in our simultaneously acquired ASL sequences. And other people who've used FPET have also seen that they see a reduction in the relationship between FPET and fMRI signals in task-related responses. And so while we need to understand this a little bit more, it does seem that the ageing trajectories of metabolic connectivity are quite a bit different to functional connectivity. In the final result I want to show you, we were just interested in the energetic costs of maintaining the metabolic network. And so in this set of analyses, what Hamish found was that the metabolic network of older adults, was less efficiently organised than younger adults. And interestingly, the older adults used more of their energy budget to maintain information transfer across this network than younger adults. And so taken together, what this means is that older adults have a smaller energy budget, but they're using a larger proportion of that budget to support a less efficient network architecture relative to younger adults. And I don't have time to go into this right now, but this was also associated with poorer cognition. So in summary, ageing is characterised by less variable and complex glucodynamics and an altered metabolic network structure. In healthy older adults, the network is less well interconnected, it's less efficient, and it costs more energy to transfer information than in younger adults. And so we think that these results and all the results I've shown you today really underscore the fundamental role that metabolism plays in supporting information transfer in the brain. And these results in particular show that unique insights can be gained by looking at metabolic connectivity and glucodynamics in ageing. So where are we now? We've got 2 minutes left. So do we know anything more about the brain than what we knew before all this work? So we knew that the structural and functional networks of the brain are shaped upon its reliance on glucose as its primary energy source. And we've capitalised on new imaging techniques using FDG-PET to see that glucose uptake does vary dynamically across time, both at rest and during tasks. And the dynamic range and complexity of this glucodynamic activity is important for cognition and ageing. Coherent glucodynamic activity lays the foundation for the metabolic network of the brain, and this seems to be predominated by connectivity in higher order networks. The metabolic network does share some similarity to the functional network, but it is also uniquely predictive as well. And lastly, metabolic connectivity is an old question in a new time. We are doing something slightly different here in that we're looking at networks within brains and with previous approaches looking across brains. However, that's not to say that we should just throw all that research away. We've learned so much about how the brain uses energy and how this is similar across groups of individuals from that research. And so we see this research as being complementary to the 40 years of research that's gone before us. And finally, metabolic connectivity is just one of a suite of molecular connectivity tools. PET is hugely powerful to understand the molecular biology of our brain in vivo. And the brain is multidimensional. Information is being transferred across multiple dimensions, including hemodynamics, which we all focus on quite a bit here, but also electrophysiological and molecular. And there are a lot of time-varying biological targets that index different components of function. So I'm really excited to see in the next 5 to 10 years dopamine networks, serotonin networks, all these other types of molecular networks that we can obtain here. So we do need to improve our methods a little bit more before that's going to be easily obtained. But I do believe that we're at the beginning of a new dawn of research to understand the molecular network architecture of the brain. And so to finish off, I do want to give my deep thanks to my PhD student, Hamish Deary, who's been instrumental in this work, Emma Liang, who's my biomedical engineer, and my mentor, Gary Egan, and all the wonderful people that have contributed to the work over the last decade. Thank you very much.
Do you want to stay at the podium?
Okay, great. Thank you so much. Your talk stimulated a lot of interest and a lot of questions, so I'll try to get to the most popular ones. So one of the questions that some of the audience members were wondering about was about the different temporal resolution, of course, that you have between fMRI and even the FPET that you described. And so did your functional connectivity analysis take this into account, the different frequency content, or do you think that some of the differences that you observed across the fMRI and PET results were due to those differences? Thanks.
That's a great question, and the very short answer is we're not sure. We do try to avoid direct comparisons because the SNR properties and the temporal resolution is so different between them. So we might do things like we compare predictive utility for cognition and age, but we don't necessarily do the direct comparison. There are questions about whether or not glucodynamics are truly as fast as what people think at a theoretical level. And so all these questions mean that we do need both technical innovation, but also innovation in our models and theories to be able to understand. So hopefully in 5 to 10 years, I'll have a more satisfactory response to that question. I think we have one up here as well.
So we have somebody at the mic.
Mm-hmm. Yeah, wonderful talk, really interesting. I'm wondering whether or not you were able to tease apart the glucose extraction fraction component, the vascular-related component, from the K3, the hexakinase metabolic component, because it might be that the former is confounding the interesting stuff, which is K3 and the hexakinase stuff.
Yeah, so we haven't done that so much. Alessandro Bertoldo's group in Padua have done a little bit of that work in probably the last 2 years or so. And they do see quite different network parameters depending on whether or not they're looking at K1, K2, K3 or SUVR. So we haven't disentangled that from our data. I think what's interesting is that some of the network topology looks quite different, particularly for K3. So you might have noticed that the connectivity between homologous brain regions in our data seems quite low. And if we have a look, it seems to be primarily in the top 5% of connected regions that we do see those connectivity profiles. And what Alessandra's group showed was that this kind of homologous connectivity seems to break down in K3. And so we are thinking that, so they use a traditional bolus approach. They do a very different analysis to what we do. But we think that possibly with our infusion approach, we might be more sensitive to that K3 parameter as opposed to K1 and K2.
Great. Okay, so we're close to running out of time, but maybe I'll just sneak one more in. You had mentioned, the differences between PET and fMRI. fMRI obviously has a vascular component, but some of the audience members were wondering about the vascular contributions to the FDG PET signal.
That's a good question. I sometimes struggle with this a bit as well because I'm also not a real PET person, and so to me it seems like, you're extracting the glucose from the blood, so why does the blood not have such a big impact on it? I think ultimately it's because we know that, that the FDG, we know where the FDG is accumulating. And so because of that, we know that it's accumulating in the postsynaptic terminal, and we can use the pharmacokinetic models to link that uptake signal with the underlying glucose metabolism. And so in theory, it's less confounded by flow than bold response. However, we do have some papers where we've looked at the relationships between CBF, bold, and glucose metabolism that you'd be able to find on our website as well.
Excellent. Thank you so much. Unfortunately, we're out of time. There are many more questions, so I'm sure you'll have some eager people coming to you after the talk. So thanks so much.
Thank you very much.